I'm a physician and a scientist, and I study the human immune system. Now, when I was a senior medical student, I worked for several months in a rural hospital in East Africa. Very interesting place, tea farmers, the Kipsigis people, very beautiful and dignified people. But working in this place at my young age was very challenging to me because of the overwhelming burden of infectious diseases in young children there. And I had just learned to put on my white coat and really my professional demeanor as a physician in training. But an event happened very early in my time there that broke through my shield and shattered my world. So I was working on the ward, the inpatient ward, and every day we had about a dozen children referred from the clinic for admission to be admitted and treated in hospital. And I was along this hall, the corridor, and there were about a dozen families waiting to be seen. And as I came in, a man tugged gently at my coat and said, "Sir, would you see my child?" And I turned to him thinking to say, "You really need to wait your turn in line, sir." But when I looked over, I saw his wife was holding a small child, and the child was limp and somewhat blue looking, and this was very disturbing. So we moved the family very quickly into our procedure room, which was just really a closet. And the mother and father and I went in with the nurse who was the translator. Now, when a doctor sees you at an exam, the first thing they typically do is take vital signs, and the first of those is the pulse. But in a child, it's actually easier to use your stethoscope to just count the heartbeat. So I put my stethoscope on, and I had the child, and I was really going to just count the heartbeats. And I was having trouble with my stethoscope, and they toggle back and forth, so I checked them and just to make sure I had it in the right place, and I'm listening, and I'm still having silence. And then I realized I'm having silence because the child does not have a heartbeat. And at that moment, in that place, with no ICU, no life support, all I could do was to hand the child back to the mother and say, "I'm so sorry, your child is dead." And that mother let out a wail that filled the entire room and the soul of everyone who was there at the time. And something broke in my heart that day. I can't forget that moment. It stuck with me forever. And I was experienced in part by proximity the sadness and loneliness that parents feel when they experience the death of a child. Sometimes when I would put my own child to sleep, I would kiss them and think about the child that died in my arms. And so I vowed in my bed that night, crying, that I would use my life and career to do something about infectious diseases, especially for the most vulnerable among us. And I would ask you, when you think about what's happened in history, who is the more formidable foe? Is it human beings or pathogens? Now, you know from your history books about the Black Plague or bubonic plague, starting about 1340. At peak, about 2 million people a year died. This is a painting from Bruegel from the 16th century. This went on a very long time. And I think most of us think of the Black Plague as the most impressive and horrific epidemic that happened in history, and certainly it was horrible. But this seems long ago and far away, like a fairy tale of sorts, almost like it didn't happen. But I want you to remember that there are people living today who were alive in 1918 when the 1918 influenza pandemic occurred, and 50 million people died in 1 year due to that flu virus. Interestingly, people who were in their 20s and 30s were very susceptible during this pandemic, as well as in the pandemics in 1957, 68, and so on. This is a photograph from a makeshift infirmary in a barracks in Kansas in the US during the pandemic. 5% of the US population died that year, 50 million people in the world, which far exceeds all of the deaths in the World War that was going on at the time. And so this is in the memory of people who are living today. And if 50 million people doesn't impress you, you may not realize 500 million people died of smallpox in recorded history. Now, smallpox is a horrific disease. It goes in every organ of your body, including your skin, and that's what you see, these pocks. And a third of the people who are infected die. Now, smallpox is our first major victory against an infectious foe. And how did we do it? We did it with vaccination. We used a weakened virus in a solution and used a little forked needle and took that needle and we pricked the skin, usually the skin of the arm, 15 times. This video is actually me getting my last smallpox vaccine because we work with similar viruses in the lab. So I'm well protected against smallpox if it ever comes back. But the thing is, smallpox does not circulate in the earth anymore because we eradicated it. So we went from 500 million people dying to no one dying. Not a single person is infected with smallpox. And this is a major victory. But you have to realize this only happened within the last, it's less than 40 years ago, and we've only done it once. And it may seem like a very crude way to fight, but this was the magic that allowed us to eradicate infectious disease, and we have not been able to do it again. Now, the WHO declared that smallpox was eradicated in 1980, and if you're a millennial, 1980 seems long ago and far away and kind of like a fairy tale. But if you've been watching the news, this keeps happening. 1997, bird flu, H5N1 avian influenza, SARS 2003 coming out in Asia, very virulent virus. We just lived through the 2009 pandemic H1N1 flu. Middle Eastern respiratory syndrome virus in Saudi Arabia, very virulent virus. Chikungunya, 1 million people in the Caribbean were infected in 1 year in 2014 with debilitating arthritis. You'll remember watching the news with the horrific outbreak of Ebola in West Africa, even came to the shores of the US. And last year, Zika virus sweeping across South America, millions of people being infected, including pregnant women, and the mosquito-borne virus is transmitted to pregnant women, passes to the fetus, causing microcephaly, fetal damage, and death. So this stuff is happening every year around us. And I want you to think, what are we going to do about this? What is the next step? What are our defenses? And I want you to just think about your own body. How do you think about your body and your health? Do you feel healthy and powerful and resistant? Or do you think of yourself as somewhat vulnerable and susceptible and at risk? And when you think about the entire human race, are we resilient and are we going to pass through all these things? Or is there some fear and a realistic fear that the entire human race could be eradicated by one of these pandemics? And I think you would be justified, given what I just showed you, to be concerned about that. So the question is, what are we going to do? Now, when people think of their body and their health, they often talk about system. They say, well, my system was run down and I got a cold last week. Or I'd really like to eat better and sleep more, build my system up and be strong. Well, what are you talking about when you talk about a system? What is that word? What you're really referring to is your immune system. Your immune system is your defense system that wards off or cures the infections after they happen in you. And I want you to think about, where is that immune system? The immune system traffics in all parts of your body, and it does that by living in your blood. So I wonder if you ever wondered what is in blood. Maybe you've never looked inside blood, so we're going to look today. Welcome to my clinic. I am actually both the physician and the patient today. And I'm going to draw my own blood. Ow. We'll just get a little drop of blood going here. So, a drop of blood is just a tiny little volume, but there's amazing. Let's look and see what we've got there. So I have this microscope is wireless, and my friends are going to project me up here, I think, live. And let's see, I've dyed this blood with a blue dye. There we go. Wow, that's pretty amazing. So the little dark brown ones here, let me focus this here, are the red blood cells, and they carry the life-sustaining oxygen. And the little ones in the center that are blue, they're called the white blood cells. If I didn't put a stain, you wouldn't see them. But see those two little blue guys? And then let's see if we can find some more. Wow, there's some more. So those are the white blood cells. And Ted, fortunately, has painted the logos for us today, so you'll remember this. The red blood cells are on the left, and the white blood cells on the right. So whenever you want to see that logo, you'll remember the two cells of the blood. So when I see these little blue cells, there's a lot of them in your blood. That's just a tiny drop of blood, and yet we see thousands and thousands of cells. But they all sort of look the same, and I've always wondered how many different cells are there. And really the question is, do we have enough tools to defend ourselves against all the millions of microorganisms around there? Is this enough? These little blue dots that are floating around in my drops of blood, how many do we have? How many different ones we have? And so I wanted to do a counting exercise and figure out how many parts of the body there are in the immune system. So when we think about parts, scientists usually these days are talking about genes, because in genes, the genetic material encode all your parts. So we're going to go back to the slides, and I'm going to give you a little genetics class. The Human Genome Project was one of the major science projects that's been done in the last several decades. So scientists spent about $3 billion back in the day and took blood and other body parts and sequenced all of the genes in the human genome. One of the amazing things that came out of that project was that there are only about 25,000 genes encoding your whole body. All your body tissues and organs are encoded by just 25,000 genes, actually less than that. But that number seems so small and not large enough to deal with all the microorganisms that we're going to face in our life. So it seemed a little scary. But what we didn't tell people was that we sort of avoided the immune system genes because there were so many of them, we were not able to get to them with the technology at the time. And the reason is the molecules in your immune system that help you fight things like viruses are called antibodies, and the antibodies are encoded by combinations of genes. So you have these large buckets of genes. There's 3 main buckets or groups, and to make an antibody, you choose one from each bucket and you put them together. And by combining these things, we get this amazing diversity. Combinatorial diversity is what we term it. And by doing that, we already have an immune system collection that's as big as all the other genes in your body. But when we put them together, they don't go just end to end. They're put together with little linkers or spacers. And these spacers are near infinite. They're like having 2 wild cards in your hand of cards if you were playing cards. And by doing that, we end up with 10 to the 11th possibilities of antibodies we can make. Now we're talking real numbers and real diversity. And then finally, we have one more trick in our immune system, and that is in our antibody genes, our defense genes, we allow those genes to mutate when they see an organism. And so we get little point mutations, so we make new genes that are just a little bit different from the ones we had before. Now, you wouldn't want to do this in any of your other genes because you'd end up with cancer. But in the immune system, this is encouraged and allowed. And by doing that, we get numbers like 10 to the 18th are possible in your body. And I was really excited about that. You can't get your mind around what is that number, 10 to the 18th. The only thing I can say is you've heard a lot of astronomy talks at this conference. You have more potential antibodies in your body than all the stars in the galaxy. It's amazing. And so a lot of scientists, like me, have obsessive compulsive tendencies, and they like to just count things. And a lot of these star guys, you were saying, I want to go out there on the shuttle and count all these things. Well, we have more antibody genes than there are stars, and my friends and I wanted to count the 10 to the 18th genes. And how do you do that? Well, you do this with sequencing. We have instruments that determine the gene sequences. So if you were actually going to count that many, what would you do? So what I would like to do is take all the white blood cells out of your body and count every one of them and sequence them. And you're giggling, but that's actually what we do. So we bring in people to our clinic at Vanderbilt, and we put them on this instrument that's like a blood filter. We put one needle in one arm and one in the other. The blood comes out. We filter out all the white blood cells into a large bag. There's 40 billion cells we collect into these bags. And we put the red blood cells back in so that you can carry your oxygen and you walk away. You're not anemic, so you're fine. And you'd say, wow, that sounds very dangerous to remove all the white blood cells, 40 billion cells from your body. Your body is amazing. It can replenish those almost immediately. You make those back and put them back. So it's a very safe procedure. This is actually a friend and a hero of mine, Morris Ibuuchi. He's a physician. He was working in Lagos, Nigeria during the Ebola outbreak. And a person came into the country and basically lied about his situation. He had Ebola and showed up in their hospital. And Morris and several of his colleagues took care of this patient. They figured out it was Ebola, and they contained the infection within their hospital. It did not leak out in Lagos. Lagos has an unknown number of people. It's so large, it's about 15 million people. If the virus had gotten out into that high-density population, millions of people would have died. But Morris and several of his physician colleagues contained that in the hospital. Morris became infected while giving care to these patients without any specific treatment. He survived and came to Nashville and talked about his experience. And it was really just an amazing talk about his courage and persistence. He survived, he's fine now, and he came and gave us blood cells. So we've been studying people like Morris, extraordinary people who face down these pathogens. So what happens when we take all these cells out and we put them in our expensive machines and we get all these sequences? Well, DNA is encoded by 4 letters, GATC. So it's GGGCTAAATC. And it just looks like this, and it's kind of like gobbledygook. So what would you do with that? Now, if you're obsessive compulsive, you love these things, and you just kind of you sort of memorize them. How many can you memorize? And my friends and I are attempting at this time to sequence all of the antibodies on the planet in the entire human population, and to put these on a database publicly so people can use them for their own studies. Now, this is an enormous task. It's the largest genetic project ever attempted, and we've already started, and we're getting billions of sequences. It's really exciting. But what would you do other than just cataloging these things? It's just an exercise in cataloging and counting. Well, it turns out there's another revolution going on in biology. It's called synthetic genomics, or synthesizing genes. And essentially, we have little benchtop instruments that are like DNA printers. So we can type in some letters and print out the DNA. And well, so what about that? Well, if I type in the sequence of an antibody that I got from Morris's body that's for Ebola, that machine will make me that DNA. I can use that DNA as the recipe to make the antibody that he was making in his body. I can make it in the lab. So then we end up with tubes full of antibodies that are the natural antibodies from his body, but we can make them forever in unlimited quantities and give them to other people. So it's really fun to make antibodies. It's sort of like having a little Play-Doh factory, and you can make things all day long. So I'll give you some examples. I mentioned there are survivors from the 1918 pandemic. We've learned a lot of things. One is that your blood retains the memory of everything you've ever seen in your experience. So we obtained blood from 100-year-old subjects who had experienced 1918 infection, and we were able to get antibodies out of them that killed the virus. So this is a high-resolution X-ray crystallography structure, but basically you're seeing a single molecule of the flu protein in the middle, and on the top in yellow and red are antibodies from a 100-year-old person that bind to and inhibit 1918 flu. Once we learned we could do it 100 years later, everything else seemed easy. Here's another example. Michelle Barnes, an adventure traveler, went to Uganda with her husband. They were on safari, seeing amazing animals, and the guide said, "Wow, if you like these animals, you should go in Python Cave with us on the last day. This cave has 40,000 fruit bats in it, and they're amazing." And Michelle did go in that cave. This is a photograph of the roof of the cave taken by another friend, Joel Sartori. He's a National Geographic photographer, a fantastic photographer. This is the actual cave in which she entered. She went to Kampala, flew home to the Denver area, and about a week later, she nearly died. No one knew what was happening to her. She recovered of her own accord, no specific therapy, and a year later, it was discovered she had had Marburg virus infection. Marburg is a deadly cousin of Ebola. It's very similar to Ebola. So she was the only known Marburg survivor, at least in the Western Hemisphere. She talked about her experience at TEDx Boulder last year. It was very, very exciting to hear. We took her blood and made a large number of antibodies. This is one of them. So in the center is the Marburg protein from the virus, and her antibodies are shown in yellow. We isolate a lot of antibodies, and they'll actually inhibit and kill the Marburg virus. So this particular antibody, these antibodies are being developed. In fact, the United States government just invested $30 million to further develop this antibody as a therapy for the next cases of Marburg or if an outbreak occurs. Another example would be Zika virus. We worked with a person who had been infected in South America last year, got cells out, made antibodies. Here you're seeing the antibodies in red, and this is called a cryo-electron micrograph, but it's just one virus particle. It's a picture of one virus of Zika, a Zika particle, and it's decorated on the surface by the red antibodies, which are attaching to and killing the virus. So in summary, when we think back to my question, who's the bigger foe? Is it infectious diseases, or is it you and your immune system? And I hope it's obvious to you the amazing depth and complexity and beauty and glory of what is within you. The human immune system is the most deep and rich information system on the planet, and each one of you have it within you. So when you think about the future, I want you to walk boldly into your future, with caution and wisdom, of course, but be confident of that glory and complexity that is the universe within you. Thank you.